Okay, so welcome to this next video in which we are discussing acyl-biotin exchange chemistry. Okay, so remember the aim of what we are trying to do. We're trying to work out which of the proteins in our protein sample are palmitoylated and which are not. Okay, so our first step was neutralizing the free cysteine residues within the proteins within our protein sample by binding any pharmalamide to them. We then split our protein sample into two, a control and one that we were going to add hydroxylamine to. And into the one that we added hydroxylamine, what happened is that the hydroxylamine molecules cleaved the palmitoyl groups off of cysteine residues, and this meant that you then had free cysteine residues on these proteins once again, which were the cysteine residues which were originally bound to palmitic acid molecules. Okay, we've now exposed our protein samples, both of them, not just the one that we've exposed to hydroxylamine, but also the one that we've kept as a control. We exposed both of them to this molecule, 1-biotin amido, 4,4' methyl cyclohexane carboxamido butane. Okay, and this will bind to any free cysteine residues. Now, in the control, there should be no free cysteine residues because they should all be bound to any pharmalamide or palmitoyl groups. Okay, but in the one which has been exposed to hydroxylamine, there will be free cysteine residues, which are those that have now been vacated by the palmitoyl groups, and to those cysteine residues this molecule will bind. Okay, so let's have an update on what has happened to our free proteins A, B, and C. Okay, and we'll draw what is the state of these free proteins in the control, okay, and also what the state of them is in the test tube that we have added hydroxylamine to. Okay, and we've now added our BMCC molecule. Okay, so in the control, let's start with protein A. Protein A will have all of its initially free cysteines bound to any pharmalamide, and it will still have some cysteine residues which have palmitoyl groups stuck onto them. Okay, so this is the state of protein A in the control. Okay, so this is protein A. Protein B in the control. Okay, let's show protein B here. Remember, protein B was originally triangle protein. Protein B had no palmitoyl groups originally on it, so all of its cysteine residues were initially free and therefore will all have any pharmalamide bound to them. And finally, protein C was like protein B. It had no palmitoyl groups originally, so all of its cysteine residues were free and therefore will have any pharmalamide bound to them. But none of them were free at the stage when we added the BMCC, so none of them will have BMCC bound. Okay, now in the test tube which we did add hydroxylamine to, what will have happened is that the palmitoyl groups will be knocked off protein A. Okay, and this will mean that we now had two free cysteine residues here, which were free at the point when we added the BMCC. Both of these will now get BMCC bound to them. So here's a BMCC bound to this one, and here's a BMCC bound to this one. Okay, and of course the cysteine that originally was free and therefore got any pharmalamide bound to it will still have any pharmalamide bound to it. Now for both protein B and protein C, you had no palmitoyl groups originally bound to cysteines. So all of the cysteines were originally free and therefore all had any pharmalamide added onto them, and therefore were all involved in bonds already to any pharmalamide and therefore couldn't have BMCC added onto them. And this is the same for protein C over here. Right, so now what you're going to do is you're going to detect the presence of this BMCC. Now how are you going to do this? Well, you're going to detect the biotin moiety. So remember, if I show you the structure of BMCC again, It's this malamide group over here which is involved in binding to the thiol group of the cysteine residue. And the biotin group is pretty much untouched over here. Okay, so it's dangling off the side of the protein. Now, there is a protein that can bind to biotin residues, biotin groups, with very high affinity. Okay, and this protein we got from the bacterial species Streptomyces avidinii. Okay, so there is this bacterial species known as Streptomyces avidinii, and from Streptomyces avidinii we got a protein known as Strepavidin. Okay, so this is Strepavidin. Okay, and this protein, Strepavidin, is a tetramer. Okay, so I'll draw this. It's a tetramer, and it has 4 biotin binding sites, and these 4 biotin binding sites are extremely high affinity. Okay, so here this is supposed to represent the Strepavidin protein, which is a tetramer. So the 4 separate subunits are here, and I'll now draw on the 4 separate biotin binding sites. So I'll highlight these in turquoise. So 1 here, 1 here, 1 here, and 1 here. So there are 4 separate biotin binding sites. Now, if we think about protein A here, which has these BMCC molecules dangling off it, which have biotin biotin moiety within them. The biotin moiety will bind to these biotin binding sites here on the Strepavidin protein. So what we do is we create what's known as a Strepavidin column, okay, which is basically a column consisting of, which is full of beads, tiny little beads. And I don't mean these are not big beads. These are absolutely tiny beads, and these beads have been covered in Strepavidin. Okay, so basically what we do is we get a tube like so. Okay, and we fill this tube up with little beads. Okay, loads and loads of the things all stacked on top of one another, and these beads have on their surface this Strepavidin protein, this tetramer here. Okay, now what we're going to do is we're going to pour both of our samples, our control sample and our hydroxylamine sample, we're going to pour them through the Strepavidin column, and we'll have 2 separate Strepavidin columns. We'll have one for the control over here, and we'll have one for the non-control. Okay, so this is the control column over here. Right, so we'll pour control through one of these, and we'll pour the non-control through the other. Now, what should happen in principle? In principle, let's take, for example, firstly the non-control being poured through. What should happen is the protein should sort of drizzle through, and what should happen is that protein A, which has these BMCC molecules strapped onto the side of it, those should bind to the biotin binding sites on the Strepavidin proteins, which are on the beads within the Strepavidin column. Okay, so what should happen is that protein A should get stuck to the beads, basically, whereas protein B and protein C should go straight through. Now, in the control, none of them have these BMCC molecules bound to them, and therefore all of them, in principle, should go straight through. Now, of course, why are we doing a control? There would be no need for a control if it was that simple, okay? If we knew what was going to happen to the control, why did we do it? Well, the reality is that some of these proteins, and this is why I've put 2 that appear to be identical, so B and C, you might have been wondering, why is he doing B and C here? Because effectively they are highlighting the same principle. They both had no palmitoyl groups. Well, basically they're now going to diverge in their purposes. I'm going to use protein C as an example of a protein which may well bind to the beads. There might be something that the beads are made up of that the protein C might bind to. Okay, so protein C might bind to the beads, and it will do that in both the control case and also the non-control case. Okay, so now let's say what has happened. So, Firstly, protein A in the control case, okay, it did not have these BMCC molecules bound to it, okay, so it has not bound to the beads, it's not bound to the Strepavidin protein tetramer, okay, and now it's gone straight through and it won't be bound to the beads. So protein A is not bound to the beads in the control. Protein B has also gone straight through. It doesn't have BMCC molecules bound to it, so it goes straight through. Protein C, however, may well have bound to the beads, basically, let's say. So protein C is bound to the beads in this case. Now, in the non-control case, protein A has BMCC molecules bound to it. It will drizzle through and bind to the Strepavidin protein tetramer on the beads, and therefore it will remain bound to the beads. So protein A has remained bound to the beads in the non-control case. Protein B does not have BMCC molecules bound to it. It's exactly the same as in the control. It will go straight through, so it doesn't bind. Protein C, again, will just bind to the beads, even though it doesn't have BMCC molecules bound to it. Okay, right. So, what you will then do is you'll run your samples through the Strepavidin columns. Some of the proteins will bind to the beads, okay, and some of the proteins will not. Then what you need to do is wash off the beads to make sure that there aren't any proteins that are just sort of lurking in the midst of the beads and just haven't managed to flow through but aren't actually bound to the beads. So you wash off, and this makes sure that only the proteins which are actually bound to the beads are still in the Strepavidin column. And then what you'll do is get rid of all the stuff that's washed through. Okay, you'll then get a new container, put it underneath this column, and you'll then do what's known as an elution. Okay, the next step is what's called an elution. So an elution is basically when you stop the proteins from binding to these beads anymore. So you're going to resolubilize all of the proteins that are bound to the beads, and they will now move through, basically. So elution is when you separate the proteins that originally bound to the beads from the beads, okay, so that they can now flow through. And you will collect the proteins in both cases, both the control and the non-control. You will collect the proteins that come through when you elute. Okay, so what do you use to elute? Well, basically, you usually use a molecule known as EDTA. Okay, so let me show you the structure of EDTA, because it's a very nice molecule again. So we've had malamide, and now we're going to have EDTA. So EDTA stands for ethylene, so ethylene, and then diamine, okay, diamino, tetraacetic acid. Okay, so E is for ethylene, D is for diamino, and then the T is for tetra, and then the A, and it should all be one word, but I've run out of space, is for acetic acid. Okay, so this again is a beautifully symmetric molecule. Okay, so we'll start off with the ethylene, which is this 2-carbon molecule here. Okay, so here's the ethylene. Then we need the diamino, so I'll put on 2 nitrogens, but these are not pure amino groups anymore. They don't just have 2 hydrogens coming off. Instead, we have tetraacetic acid. So basically, you have 4 acetic acid groups, which is just ethanoic acid. Acetic acid is the old name for ethanoic acid. It's the acid which is within vinegar. Okay, so you have 4 acetic acid groups coming off. So here's the second coming here. Okay, and then 2 more off here. Okay, and that's why it's so beautifully symmetric. So here again is the carboxylic acid group of this acetic acid molecule, and then finally the final acetic acid molecule is here. Here's the methylene group, and then the carboxylic acid group over here. Okay, so this is ethylene diamino tetraacetic acid, or EDTA for short, and this is often used to elute the proteins from the beads. So you will pour the EDTA through these columns, okay, and it will break the proteins off from the beads, and they will then move through the column, and you'll collect them at the bottom. And obviously you'll collect them in a different container to what you originally collected, the stuff that you were just pouring through the column when you were just pouring your samples through the column. Okay, so you'll then have a flask which will contain a solution containing all of the proteins that bound to each of the columns. So you'll have a flask for the control over here, and you'll have a flask for the non-control. Okay, now what you will do is you will analyze what proteins are within these flasks, okay? And you could do this, for instance, using mass spectrometry. Okay, so you will find out which proteins are within there. So basically, mass spectrometry will tell us that protein A and protein C are in here, and that protein C is in here. Okay, and then what we will be able to deduce is that protein A is the only one which contain palmitoyl groups, because it's the ones which are present in here and not present in here. Okay, so it's the ones that are present in here subtract off the ones that are also present in here that originally had palmitoyl groups on, because it is those that only bound to the beads because they had palmitoyl groups put on. Okay, so protein C bound to the beads whether it had the BMCC molecule stuck onto it or not, basically. Okay, so let's think this through. So, basically, we started off with these three proteins, A, B, and C, and protein A was the only one with palmitoyl groups stuck on. Okay, now, the way we have set up this acyl-biotin exchange chemistry, what you will do is you replace palmitoyl groups with this BMCC molecule, which then has the biotin bound to it. Okay, and basically, this biotin will bind to the Strepavidin protein tetramer on the surface of these beads. Okay. On the other hand, there are some proteins which will just bind to the beads whether or not they have BMCC molecules bound to them, and those will bind both in the control and the non-control, whereas the proteins which are only binding because they have the BMCC molecule added onto them, which indicates that they originally had palmitoyl groups on them, those will only bind in the non-control. Okay, so we're looking for which proteins bind in the non-control but don't bind in the control, because those are the ones which only bind to the beads because they have BMCC molecules added onto them, and therefore those are the proteins which originally had palmitoyl groups bound. The ones which bind in both cases are just binding to the beads anyway, because we know in the control none of these have BMCC bound to them, because none of them have free cysteine residues when you added the BMCC, because we'd already neutralized all of the free cysteine residues with the N-ethylamide, and we hadn't knocked any palmitoyl groups off because we hadn't added hydroxylamine, so the BMCC cannot have bound to the proteins, basically. Okay, so that's the way in which acyl-biotin exchange chemistry can be used to determine which proteins in a sample of proteins have palmitoyl groups stuck on the side of them.